All right, everyone, welcome. We're about to do a high-yield review of what I like to call the metabolic engines of gram-negative pathogens. We're not just going to memorize facts today. Now, we're going to get to the why. Why do these bugs act the way they do, from what we see on a petri dish all the way to the patient's bedside? This is the kind of biochemistry that really can make or break your clinical decisions. So let's meet our two main players for this story. In one corner, you have the famous Escherichia coli. It's a true metabolic generalist, totally happy to ferment its way through life if it has to. And in the other corner, we have Pseudomonas aeruginosa, a very strict, very specialized aerobe. You see, their entire game plan, their entire survival strategy is dictated by one thing: how they make energy. And trust me, understanding this difference is absolutely crucial for your exams.
So here's our game plan. First, we'll compare their core strategies. Then we're going to dissect the glucose pathways that really define them. After that, we'll see how those pathways leave a tangible footprint in the lab. Then, and this is critical, we'll explore how their metabolism affects whether or not our antibiotics work. And finally, we'll wrap it all up in a high-yield playbook.
Okay, let's dive into strategy. We're about to see how the presence, or absence, of a single enzyme forces these two pathogens down completely different evolutionary and clinical paths. It's pretty amazing. Now let's run the glucose gauntlet. How a bacterium decides to break down a simple sugar molecule, well, that defines its entire lifestyle. We're talking everything from basic energy production to its ability to cause disease.
So most bacteria you know and love, like our friend E. coli, use the classic pathway, the Emden-Meyerhoff-Parnas pathway. You just know it as glycolysis. The formula is pretty simple, right? You got to invest 2 ATP to prime the pump, you split that 6-carbon sugar in half, and then you hit the payoff phase. The net result? A modest 2 ATP and 2 NADH. This is the absolute workhorse pathway for any bug that ferments.
But here's the twist in the story. Pseudomonas is missing one single, absolutely crucial enzyme, phosphofructokinase, PFK. You can think of PFK as the pacemaker of glycolysis. It sets the whole tempo. Without it, the EMP pathway is a complete dead end. This forces Pseudomonas to find a clever and entirely different workaround.
And that workaround is the Entner-Duderoff pathway, or ED pathway. It just bypasses that broken PFK step entirely. It starts with an oxidation reaction, and right away, boom, it generates NADPH. Now look closely at the yield here. You only get 1 ATP, but you get both NADH, which is like the cell's cache for energy, and you get NADPH, which is the raw material for building new parts and, importantly, defending against attack. This difference is not trivial. It is a crucial distinction.
Now, running alongside these pathways is the pentose phosphate pathway. And look, the PPP isn't really about energy. It's about building blocks. It has two main jobs. Job number one, generate a ton of NADPH. This is the cell's primary shield against the oxidative stress our immune cells throw at it. Job number two, produce ribose 5-phosphate. That's the sugar backbone for every single nucleotide in its DNA and RNA. So for a pathogen that's trying to divide rapidly, this pathway is completely non-negotiable.
Okay, time for a quick pop quiz to lock this in. Which pathway's main job is making that NADPH, the stuff that's absolutely essential for fighting off the oxidative stress from our immune cells? You got it, it's the pentose phosphate pathway. That's the cell's shield. All right, next question. Both the EMP pathway and the TCA cycle, they generate loads of which key cofactor that's destined for the electron transport chain to make those big ATP payoffs? Yep. That's NADH. It's the cell's main energy currency. And for the final question, what makes the ED pathway's output so special, so unique? It's the only one of these that generates both NADH for energy and NADPH for building, all in one streamlined process. That's the metabolic signature of an aerobic specialist right there.
This table just boils it all down perfectly. Look, EMP gets you 2 ATP and 2 NADH. The ED pathway gives you half the ATP, but you get one of each type of cofactor. And the PPP, it says, forget ATP, I'm going to max out on NADPH. The key takeaway here is that a cell's choice of pathway is a strategic trade-off. It's balancing immediate energy needs against its long-term potential for building and defense.
So that's the biochemistry. But how does this actually show up in the real world? Let's decode the metabolic footprint. We're about to see how these invisible chemical reactions leave very visible, very predictable clues for us in the diagnostic lab.
And the TSI slant just illustrates this difference beautifully. On the left, E. coli uses its EMP pathway to go to town on the sugars, fermenting them and producing a ton of acid. The pH indicator in the tube turns the whole thing bright yellow. That's a classic A over A reaction. But Pseudomonas, as a non-fermenter, it can't even touch the sugars. So instead, it metabolizes the peptones in the aerobic part of the slant, producing alkaline byproducts that turn it red. The bottom of the tube, the butt, stays unchanged. A classic K over K reaction. See? Two different metabolisms, two totally different results.
Now let's talk about the citrate test. A positive result, like the bright blue you see with something like Klebsiella, doesn't just mean the bug has a TCA cycle. I mean, most aerobes do. What it specifically means is that the bug has the front door to bring the citrate in. It has a special transporter, a citrate permease, to get that citrate from the media into the cell. E. coli, for instance, usually lacks the store, so even though it has a TCA cycle, it tests negative. No entry, no positive test.
You know, some of these metabolic tests point directly to how a bug causes disease. Take Proteus and its incredibly rapid urease activity. It takes urea and splits it into ammonia, which is a strong base. So in the urinary tract, this just jacks up the urine pH, causing minerals to crash out of solution and form these massive, dangerous struvite kidney stones. The enzyme isn't just a diagnostic marker, it's a weapon.
Okay, now for what might be the most clinically important point of this whole talk. Metabolism dictates lethality. Antibiotics are not magic bullets. Their power, their ability to kill, depends entirely on the metabolic state of the bacterium they're targeting. And the perfect example of this is the aminoglycoside class of antibiotics. To get inside a bacterial cell, they need to be actively transported. And that transport is powered by the proton motive force, the PMF. You can think of the PMF as the cellular battery that gets charged up by active respiration. So an obligate aerobe like Pseudomonas has a constantly roaring PMF. Its battery is always fully charged, so it sucks up the drug. But an anaerobe, or even a facultative anaerobe hiding out in an abscess, it has a weak or non-existent PMF. Its battery is dead. The drug can't get in, and that is exactly why you never, ever use aminoglycosides for anaerobic infections.
Now, there's a newer theory that suggests many of our bactericidal drugs actually share a common kill mechanism. It goes like this. First, the drug causes some initial stress. In a desperate attempt to make enough energy to repair the damage, the cell panics and revs up its TCA cycle and its whole respiratory chain. But this hyperactive state is unstable. It starts to leak electrons. And those electrons create a lethal storm of reactive oxygen species, ROS, that basically just shreds the cell from the inside out. It's like the drug pushes the accelerator to the floor, but it's the bug's own metabolism that crashes the car.
This concept also explains why sometimes our best drugs just fail. For a beta-lactam to be lethal, it has to create what's called a futile cycle. It blocks the cell from building its wall, while the cell's own autolysins keep tearing the wall down. But what about a dormant, slow-growing cell, like you'd find in a biofilm? It shut down its autolysin activity. So the drug can bind its target, sure, but without that active degradation, the cell doesn't pop. It becomes tolerant. It's not genetically resistant, it's just metabolically asleep.
Okay, let's bring it all home now with the board review playbook. This is the high-yield synthesis, the stuff you absolutely need to know to walk into your exam feeling confident.
This right here is the money slide. If you're gonna take a screenshot of anything, this is it. Let's run it down. E. coli, the generalist, uses the EMP pathway. It's oxidized negative. It's an A over A on TSI. Its PMF state is variable, which means it's only susceptible to aminoglycosides when it's actively respiring. Now Pseudomonas, the specialist, uses the Antner-Deuteroff pathway. It's oxidized positive. It's a K over K on TSI. And as an obligate aerobe, its PMF is always high, always on, making it highly susceptible to aminoglycosides. You need to know this table. It connects every single concept we've talked about today. Ultimately, it all comes back to this one idea. Our goal as clinicians and scientists isn't just to put a name to a pathogen. It's to understand how it lives, how it breathes, how it survives, to understand its metabolic vitality, and then use that very engine of survival against it. And that leaves us with a final, kind of provocative thought to chew on. If dormant bacteria are tolerant to our drugs because their metabolism is shut down, could the future of treatment involve intentionally waking them up? Just think about it. Could the cure for a resistant infection actually be a carefully prescribed dose of sugar, used to jumpstart their metabolism and make them vulnerable to our antibiotics once again? It's something to think about.